The hygiene hypothesis states that a lack of early childhood exposure to infectious agents, microorganisms, and parasites increases susceptibility to allergies and other inflammatory illnesses due to immune deficiency. The hygiene hypothesis relates to our microbiome. This is Bob. He is a dangerous parasite that can make a person very sick. This is Kevin. He is a helpful and necessary bacteria that is part of the body's microbiome. The microbiome is a group of countless bacteria and microorganisms, just like Kevin. They help carry out bodily functions such as digestion. They also assist the immune system in fighting off illnesses. A rich and diverse microbiome is good for the body.
The microbiome can be altered by one's lifestyle. Childhood environment has a large impact on your microbiome later in life. When parents expose their children to dirt and germs and let them play outside and expose themselves to many environments, it allows the body to build up a strong immunity through a large microbiome. The use of antimicrobial products such as hand sanitizer and antibiotics can also affect the microbiome. These products do not target dangerous pathogens specifically. Instead, they kill all bacteria, good or bad. This can negatively impact the microbiome, which will lower the body's ability to fight off bacteria and germs. Lifestyle choices as an adult also affect the microbiome. For example, a doctor in a healthcare setting will be forced to live an extremely clean lifestyle. They are always washing their hands and using hand sanitizer. Careers involved with dirty, outdoorsy work could oppositely affect the microbiome and lead to a stronger immune system. Life in an urban setting might decrease exposure to dirt and germs, whereas a more rural home will allow constant exposure to a diverse crop of plants and animals. After learning all of this crazy information about the microbiome, we wanted to conduct our own experiment to discover what else might have an impact on the microbiome and how we might use this information to prevent inflammatory diseases.
In order to analyze the differences in our microbiomes, we conducted an experiment in which we took cotton swabs of either our forearms or belly buttons in order to analyze them for microorganisms. We ran the samples through a machine that shone light through each sample. Large amounts of microbes would block light. The machine used the amount of light to calculate a number for us. We then used this number to determine how much of each species of biomolecule were present. These different food sources allowed us to differentiate between each microbe, just like everyone has their favorite food. We tested 31 different substrates as well as a water control. If the water control blocked more or equal amounts of light as one of the substrate slots, we said there was not any of that biomolecule present. This was crucial for us to determine the species richness of each person's belly button or forearm. Species richness is used in all three of our graphs as the dependent variable.
Before beginning our experiment, we hypothesized that bathing frequency, type of birth, and presence of a belly button ring would all have significant effects on our microbiomes.
Here's a graph which shows the relationship between species richness and bathing frequency. As shown on the graph, species richness is on the y-axis and frequency of bathing is on the x-axis. There is no significant difference in the species richness when comparing different frequencies of bathing. No correlation can be made because of all the different bathing habits have about the same average and range of species richness.
This graph shows the relationship between species richness and whether or not the participant had a belly button piercing. Presence of belly button piercing is located on the x-axis, and species richness is on the y-axis. As you can see in the jitter plot, there were many more participants who did not have belly button piercings. Since there was no significant difference in the average species richness for participants with belly button piercings compared to those without, no correlation could be made.
This graph shows the relationship between species richness and type of birth. You can see that species richness is on the y-axis and type of birth on the x. As you can see, there was not a significant difference in species richness between vaginal birth, not sure, or C-section. But there does seem to be a significant difference in the range of species richness for vaginal births. The graphs show a broader range. This is interesting because it may reflect the varying microbiomes of the mothers, since vaginally born babies are exposed to their mothers' microbiomes at birth.
Unfortunately, our experiment did not lead to any significant differences in species richness. We did, however, have a wide range of species richness present in our samples. This is confirmed by papers which we have read that continually state that each person has their own microbiome and no two are the same. According to Okada et al., when describing the results of their paper on the hygiene hypothesis and our immune system, Taken together, these data open new therapeutic perspectives in the prevention of autoimmune and allergic diseases. This study is important and provides a good structure for possible future studies of the microbiome.
Findings about the microbiome are very important to the general public because they allow us to further understand the microbiome and find ways to remain healthy. Results that support the hygiene hypothesis show us how to raise our kids, what type of environment they should grow up in, and how we should allow them to play. It also provides a basis for understanding the effect of antimicrobial soaps and excessive hand washing. The microbiome allows us to thrive in the ever-changing world, and without it, the immune system might not be able to fight off foreign invaders, and could lead to a lower standard of human health. If we are able to further understanding and share knowledge, we can promote ideas that support microbiomes and hopefully help prevent the spread of disease.